Hello everyone, this is your instructor Kwame Ishen, and this lecture is part 1 of our microbial genetics lecture. The objectives of this lecture is to be able to explain what genetics, genome, chromosomes, genes, genetic code, genotype, phenotype, and genomics, what these terms mean, and be able to differentiate one from the other. And then we'll also look at DNA, the importance of it, the structure and how it's connected to the function, and then DNA replication, and be able to describe transcription and translation as well. Genetics is the study of genes and how information is stored and transferred from one generation to the next generation, and how proteins are made as well. There are certain terminologies that we generally used in genetics, and we have genome, which is the genetic information in a cell. So in terms of prokaryotes, you're talking about the chromosomes and the plasmid. In eukaryote, you're talking about the chromosome and the mitochondria genes as well. So the entire genetic information in a cell is the genome. And chromosomes are made up of structures called DNA, and these DNA contain the information that forms genes, and that information that is constituted in genes is what is passed on from one generation to another, and also the information that codes for specific proteins and RNAs. So pictorially, this is a genome, this is a cell. So if this is a cell, then that's a genome, the entire genetic composition. And the genome is made up of these chromosomes. So this is a chromosome. Okay, so you have a chromosome here. The chromosome is made up of DNA, and the DNA has segments which are called genes. And these segments will code for specific proteins. Okay, so that's how the interconnection is. Now, the genes that are contained in a cell may not be expressed at all, may be expressed partially, may be expressed together, or may not be expressed in the presence of another. So, for instance, if we look at this on face value, you have a pair of chromosomes. So these are chromatids that form a chromosome. So let's say it's in a cell, and these codes for color. So you have brown color, and then you have black color. In the end, we see that the organism is showing brown color. So what it means is that brown is dominant over black, and that in the presence of brown, black is not expressed. Okay. So this information that is in the gene is the genotype. It's the genotype of the cell that will make up the organism. And the ones that we see, the observable traits, are the phenotype. So that's the phenotype of the organism. If you look at DNA structure, DNA is composed of nucleotides. DNA is a macromolecule. It's made up of nucleotides, which are the monomers. And each nucleotide is made up of these 3 components. A nucleotide is made up of a 5-carbon sugar. So that's the 5-carbon sugar. Made up of a phosphate group. That's the phosphate group. And it's made up of a nitrogenous base. and that's the nitrogenous base. Okay. The nitrogenous bases differ between DNA and RNA, but since we're sticking to DNA for now, the nitrogenous bases are adenine, cytosine, guanine, and thymine. Note this, that for the 5 carbon base in DNA, the second carbon, this is carbon 1, 2, 3, 4, and 5, the second carbon has hydrogen atom attached to it. For RNA, it has OH attached to it. So that's one major difference between the sugars. So what's one is deoxyribose, there is no oxygen here, deoxy, and this is a ribose sugar for RNA. So the nucleotides can be joined together, and that is an example of polymerization. You're forming a polymer. Remember dehydration synthesis from previous lectures. So you have two nucleotides that form a bond known as a phosphodiester bond, and in the process water is released. So you have the 3 carbon attaching to the phosphates, and it's the phosphate that is connected to the 5 carbon of the other nucleotide. And this is how it appears. So you have the backbone made up of phosphodiester bonds. That's a phosphodiester bonds. So that's 5 here, that's 3 here. I'll explain why we have 5 and 3. And then you have the nitrogenous bases that are attached to the 5 carbon sugar. DNA is a double helix. What it means is that DNA has 2 strands. So you see this red strand and the blue strand, like a ladder. And then you have these racks that are connecting the ladder to each other. And then it's twisted together like a spring. So it's a double helix. This structure was discovered by 4 people. We have Wilson and Rosalind, and what they did is that they used X-ray to figure out predict the shape of DNA. That is a double helix. And then Watson and Crick used that information to actually predict the shape and the base pairs that will come together to form the DNA structure. Watson, Crick, and Wilson ended up winning the Nobel Prize in 1962. Rosalind had died in 52, so she wasn't eligible for nomination by then. So DNA has 4 bases. We have the purines and the pyrimidines. The purines are adenine and guanine, and the pyrimidines are cytosine and thymine. The difference between the two is that the purines have 2 rings. So adenine has 2 carbon rings, guanine has 2 carbon rings, and the pyrimidines, they have just 1 carbon rings. Okay. And note that adenine will always pair up with thymine. Adenine will always pair up with thymine, and cytosine will always pair up with guanine. So the structure of DNA described is that it's made up of 2 strands that are complementary to each other, because only certain base pairs can pair together. If we have the information for one strand, if you have information for this strand, we can actually predict what the other strand is going to be. So in this case, you have thymine. Thymine always will pair up with adenine, so pair up with adenine. Adenine will always pair up with thymine. Cytosine will always pair up with guanine, like that. So we say they are complementary to each other. And it's important that during cell division, duplication, when DNA is duplicated before cells divide, this duplication is done accurately. Then you have the other factor in terms of the structure of DNA is the fact that it is anti-parallel. So it's complementary, it's anti-parallel. Anti-parallel in that you have 5' end that lies at the same side as a 3' end for the other strand. So for one strand you have 5' end and 3' end. The other you have 3' end and 5' end. And the reason why we have those numbers is this. The 5 carbon sugar is labeled. So this is 1 carbon, that's 2, that's 3, that's 4, and that's 5. Okay. Note that the bond is formed between the 3' and the 5', like this, and the 5'. So you have a 3' end here, and you have the 5' end here. So for the opposites, the other strand, you have 3' and 5' like that. So they are anti-para. So they are complementary and they are anti-para. So that if you are given, you know how they pair up. If you're given one strand as a 5' to 3', you can actually create the other prime. You can predict how the other prime is going to be like. So it's going to be 5', a thymine will pair up with adenine, and cytosine with guanine, like that. So you should be able to do that. And so try your hands on this exercise, okay? So that brings us to the genetic code. The arrangement of the genes in a DNA structure and the predictability in terms of the different strands, in terms of base pairing, leads to certain combinations. So you have the linear sequence of the bases that will provide information and tell the cell what proteins to synthesize. And we have the genetic code, which is a set of rules that we have used the information that we have gotten from the cells to actually decipher that if these sets of genes are transcribed together, then we'll have these proteins. Okay. So moving from the genes to the proteins or amino acids is what we call the central dogma. There are steps involved in that. If we look at the eukaryotic chromosome structure, it is a bit different in terms of how it's packaged. So you have 2 copies that are present, or you may have a single copy like this, chromatid, or you may have chromatids that are joined together at the centromere. Now these are wrapped together around proteins called histones and other proteins as well, and you have a number of non-coding regions. In bacteria chromosome structure, these are found in the cytoplasm. For eukaryotes, they'll be found in the nucleus. And you always have a single secular copy. Okay, so you have a single secular copy. During cell division, it's duplicated. But generally, because you have, for instance, 1,000 times longer base pairs compared to the size of the organism, there is what we call supercoiling. It's coiled several times in there. And this supercoiling helps a lot of DNA genetic material to be packaged into a cell. The sequencing, that is knowing the base pair sequence and being able to characterize the molecules that make up the genome, is known as genomics. So we have tried to sequence the genetic sequences in the genome of bacteria, and so many of them has been done. In 1984, we started the Human Genome Project to sequence the entire genetic sequences in the cells of humans, the different cells of humans, and this was completed in 2003. So back to the central dogma again. The central dogma, you're moving from the genes, that is from the DNA, to proteins. Now the steps that are involved are these. You have DNA that contains all the information needed to make the proteins, and this DNA would then be copied into a messenger RNA. We call it transcription, and then a messenger RNA would then be copied into a protein or translated or interpreted into protein molecules. Before this is done, cells will divide. Before cells divide, there's replication that takes place, DNA replication. Now, all these involves different proteins, enzymes, and carriers, and other structures that are involved in this process. So the flow information, again, there's the DNA replication, that's separate. But from DNA, there is transcription to RNA, from RNA to protein, and that is the central dogma. So DNA replication basically happens during cell division. And before the cell divide, the genetic material will duplicate, make a copy of itself, and then it will be distributed equally among the offsprings. Now, what you need to note at this stage is that each original strand is a template. Each original strand of the DNA of the parent cell will serve as a template for the offspring. And this is what I mean by that. There were three theories that were proposed as to how DNA copying or DNA replication happens. Now, the first one was that for the old DNA molecules that are found in the parent cells, that those are maintained in one of the offsprings, so that you have this parent. Okay, so this is parent. This is parent, and it would then be maintained in one offspring, so that the one offspring will be the exact copy of the parent, and then new copies are made and they go into a separate cell. Okay. The other was that each of them will become a template and then be copied. A complementary strand of each of them will be made, and then they will enter into new cells separately. And then there is a third model, that is the dispersive model. So there's the conservative, there's a semi-conservative, then there's the dispersive. The dispersive model says that the two, in terms of the new and the old, are mixed together. So you have old genetic material, new genetic material, like that they're all mixed together. What we have noticed is that it is this model that actually is the correct one. The semi-conservative model is the correct one, so that you would have original parent strand and each of them becomes a copy. So they will separate. So you have this and this. These are the old strand. Copies will be made, T, A, T, G, C, complementary and antiparallel. And then that the offspring will contain half the genetic material from the parent. Okay. The same concept that is done here. That's the parent, the offspring will have. And the next generation, you would notice that half of the offsprings will have half of the grandparent genetic material, and the other half would have just copies of the parent. So this is a one parent, G1, that is copied, and then you have G2 here. So that only half of the chromosomes are that of the parent, but then you would have a quarter of it that would be that of the grandparent. So you have these two having half of their chromosomes being that of the grandparent. So there are steps involved in replication. You have a point of origin where replication begins. That's the origin of replication. In prokaryotes, there is only one point of origin. And then in eukaryotes, you have multiple origins of replication. You have different steps that are involved. There's unwinding and unzipping. And then you have the primers that have to attach. And then you have replication that takes place. These enzymes then play a crucial role in the process. So there's the steps of replication. You have uncoiling. remember there is supercoiling of the chromosomes or the DNA. And this has to be uncoiled to make the base pairs available for copying to be done. And this uncoiling is done by DNA gyrase or DNA topoisomerase. Okay, topoisomerase. Isomerase. SE. Okay. Now, once that supercoiling has been removed, DNA still appears as a double helix. It appears still as a double helix like that, and so it has to be unwounded. It has to be unwounded, and then it has to be unzipped, and that process is catalyzed by DNA helicase. Remember, enzymes end in ASE, so DNA helicase does that. You then have an origin of replication along a replication fork. So this will be the origin of replication. This will be the replication fork. So because it's opening the DNA in this direction. It's opening the DNA in this direction. That's the replication fork. You will then have an enzyme known as DNA polymerase. So it's going to be a polymer. It's going to add nucleotides, DNA nucleotides. And DNA polymerase will then synthesize the new strand. So you have a new strand being synthesized here, another one being synthesized over here. But note this, DNA polymerase can only add nucleotides to the 3' end, only to the 3' end. So it can only elongate from 5' to 3'. So we say DNA polymerase synthesizes the new strand in a 5' to 3' direction. Okay, you have, this is the 5', there's the 3'. DNA polymerase will only add to the 3' end. So if it adds, you end up having the extra nucleotide added here. So it only elongates from the 5' to the 3' direction. This is a very important diagram that you should master. And so we're talking about replication. This is what's going on. There's replication. You have the parent DNA material. It's divided into two, but before that, double the material has to be created as replication, and then half of it will go into each offspring. But in the breakdown is this. So you have the supercoiling that is removed by topoisomerase, and then there's unwinding that is done by helicase. So helicase will do this unwinding. You have single strand binding proteins, S, B, P, single strand binding proteins. So these are the single strand binding proteins, and they will hold the strand apart so that it doesn't stick together again. This is the replication fork, and that is the direction in which the DNA is being opened up, unwounded. You need a primer, a molecule called RNA primer, to attach to where the replication has to start. And the RNA primer is synthesized by primase. So primase will then attach to where the replication should start, the new strands to be created should bind to, create an RNA primer, and it's only when an RNA primer is present that DNA polymerase can attach to the template strand, that is the parent strand, and synthesize a new strand. Okay, so polymerase then will attach and then synthesize a new strand. Remember, polymerase only synthesizes in a 5' to 3' direction. So on this particular strand, you have 3' here. So this is 5', and it can only synthesize in one direction. It's able to do that very easily. So this strand is known as the leading strand. The leading strand is the strand where DNA polymerase is able to synthesize the new strand continuously. However, if we come to this other strand, you have 5 prime here, and so this is 3 prime. Remember, DNA polymerase can only add to the 3 prime end, so the 5 prime end will be here. So on this strand, DNA polymerase cannot synthesize a continuous strand. It does it in bits and pieces. So it does a little bit of it and does a little bit of it in this direction, because this is the 5 prime to 3 prime direction. It does a little bit of it like that, in that direction. And then because of that, you have short segments of DNA, new strand of DNA that are created. And those fragments are called Okazaki fragments. Okazaki fragments. And because they are separated from each other, they need to be added together. So you need an enzyme to do that. DNA ligase does that. So DNA ligase glues together the fragments of DNA on the lagging strand. So this is known as a lagging strand. This is known as the leading strand. Okay. So go over this and make sure you understand the process. In prokaryotes, the synthesis is done in a bidirectional manner. So you have the DNA that is separated, opened up, unzipped. You have your 5 prime and 3 prime, so continuously. You have, this is the replication fork. This is where the opening is taking place. But you have the 3 prime here for the lagging strand. So it's created in bits and pieces. Okay. The green represents the primers. And so it takes place in both directions because you have 2 replication forks. You have 2 replication forks happening. The interesting thing with replication, it's amazingly accurate. And the reason is that DNA polymerase does what we call proofreading. DNA polymerase does proofreading. It's just like you write a paper and you edit it before you submit the final version. So errors do happen, but you have 1 in 10 billion base pairs. So for every 10 billion base pairs, that is when you find 1 error. It's very, very small. And that means the proofreading it's exceptionally accurate. And so it goes through all the new molecules that have been synthesized and checks and makes sure that every base pair have correctly been paired. And if they haven't been correctly paired, there's an error. It excises it, cuts it off, and puts in the right base pair. Very, very amazing. These are some differences between replication in prokaryotes and eukaryotes. And so some of that is that number of origins, as I've alluded to. You have single in prokaryotes, you have multiple in eukaryotes. Replication is way faster. You have larger nucleotides in terms of Okazaki fragments. You have smaller ones in eukaryotes. Remember we said that in prokaryotes, we know for in terms of the shape of the DNA, it's circular. Okay, it's a single copy, it's circular. So how does it divide? So there's a point of origin. So there's a point of origin, and then there's the division. So you have the DNA, 2 strands of the DNA. There's a division here. Replication would take place bidirectionally, like I've already said. And it will go until they get to the point of termination, and then it's separated from each other. And the enzyme topoisomerase will help separate the 2 loops from each other. So this brings us to the end of part 1 of our microbial genetics lecture.